If you've ever had a paper cut, or maybe bumped your elbow hard enough to see that immediate localized reaction, the heat, the redness, that throbbing feeling, you're actually witnessing something pretty incredible.
Uh-huh.
It's one of the most ancient and, well, dramatically orchestrated events in your biology. Acute inflammation.
Right. It's your body's fastest, non-negotiable first line of defense against any threat, whether that's trauma, an infection, maybe toxins.
It sounds like a rapid military mobilization almost.
It really is. And today we're taking a deep dive into the absolute foundational element of this whole response. We're talking about the instant, incredible choreography that happens inside your blood vessels.
The vascular side of things.
Exactly. Acute inflammation is fundamentally a vascular phenomenon. It's a process where your blood vessels switch gears. They go from being, routine plumbing to an emergency delivery service.
Okay.
So we're going to unpack the three fundamental actions needed for this defense. First, there are changes in the small vessel diameter, which leads to increased blood flow.
More blood getting there.
Right. Second, there's a dramatic increase in permeability. The vessel walls become leaky, basically allowing necessary proteins and defense cells, the leukocytes, to leave the circulation.
Okay, leakiness.
And third, there's the guided exit and activation of those leukocytes once they get out.
So our mission today is to trace this whole sequence, try to understand the molecular commands that are running the show, and maybe see why messing with these mechanisms is, well, both key to modern medicine, but also a source of some pretty significant therapeutic challenges.
Absolutely. But before we even get into how the flow changes, we really need to clear up one of the most common points of confusion, I think, in immunology, or maybe just basic biology.
Right.
We have to distinguish between two different types of fluid accumulation, two types of swelling, because this difference basically defines what acute inflammation is.
Ah, right. You're talking about exudate versus transudate.
Exactly. This is the critical first aha moment, you could say. Swelling or edema just means there's excess fluid outside the blood vessels in that interstitial space.
Yeah, fluid buildup.
But how it got there and what's in that fluid matters immensely.
Okay, so let's start with the simpler one, maybe. The transudate. What's that?
Right. A transudate is essentially just an ultrafiltrate of blood plasma. Think of it like really watery fluid. It has very low protein content, mostly just albumin, the commonest protein, and almost no cells or debris.
Okay, watery, low protein.
And the crucial point is it leaves the vessels without the vessel wall itself changing. Its permeability hasn't increased. The endothelial cells lining the vessel, they aren't actively opening up any gaps.
So the vessel structure is intact. Then why is fluid leaking out at all if the wall isn't leaky?
It's purely a physical problem. It's an imbalance of forces, hydrostatic or osmotic pressure.
Like plumbing pressure.
Pretty much. Think about congestive heart failure. The blood backs up in the veins, right?
Mm-hmm.
That increases the hydrostatic pressure inside the vessels, literally forcing fluid out into the tissues.
Okay, too much pressure pushing out.
Or, conversely, think about severe liver disease. The liver makes albumin. If it's not making enough, the protein concentration inside the vessel drops. That lowers the osmotic pressure that normally pulls fluid in. So fluid leaks out because there isn't enough pull keeping it inside.
Okay. So it's either too much push out or not enough pull in.
Exactly. It's like a coffee filter that's just too full, or the coffee itself is too weak, maybe. Water is being pushed through just by the weight, or it's not being held back effectively, but the actual holes in the filter paper haven't gotten any bigger.
That makes sense. It's a pressure thing, not a structure thing.
Yeah.
So then the exudate must be the opposite. It must be driven by a structural change.
Precisely. The exudate is the true defining hallmark of acute inflammation. This is fluid outside the vessels that has a high protein concentration and lots of cells and cellular debris.
Like pus.
Pus is a perfect example that's a purulent exudate, just loaded with dead neutrophils and proteins. The key is that forming this protein-rich fluid absolutely requires a physical change in the vessel wall. It demands increased vascular permeability.
The holes in the filter do get bigger.
Yes. The vessels have actively opened up to let the big, important defense materials out, things like large proteins, fibrinogen for clotting, and the entire cleanup crew of leukocytes. They need bigger gaps to get through.
So when we see that hot, red, swollen tissue around, say, an infected cut, that swelling is the exudate. It's proof the vessels have launched a full-scale active response, not just a passive pressure leak.
Exactly. That distinction, exudate signals active inflammation, transudate signals imbalance, is absolutely fundamental.
Okay, so with the exudate established as the hallmark, Let's trace the physical process inside those vessels. The instant a threat is detected, the body needs to get maximum resources to the site. This starts with vasodilation, right? Opening the pipes.
That's the very first thing. It's one of the absolute earliest manifestations, happening within seconds or minutes of the injury. Chemical signals get released by damaged cells almost instantly.
And what's the main signal?
The big one initially is histamine. We'll talk more about mediators later, but histamine hits the smooth muscle cells wrapped around the small arteries, the arterioles, causing them to relax and widen. Then this quickly opens up nearby capillary beds too.
And that widening means a huge surge of blood flow into that area,
Mm.
which explains two of those classic cardinal signs of inflammation we learn about.
Right. Redness, the rubor, and heat, the calor. It's literally the flush of hot, oxygenated blood rushing to the scene because the pipes are wider.
Why is that initial flood so important, just getting blood there fast?
Well, speed is part of it, yes. It's the delivery step. You need to get the blood carrying all the defense components, plasma proteins, clotting factors, leukocytes there quickly and in large volumes.
Okay.
But it's also about distribution. By widening the arterioles, it forces open downstream capillary beds that might normally be closed or have low flow. This maximizes the surface area, the interface between blood and tissue, which is crucial for the next steps where things need to leave the blood.
So it's like opening up all the local roads, not just the main highway.
Exactly. But interestingly, you mentioned that this initial rush is quickly followed by something that seems, well, contradictory. Stasis. Things slow down.
Yeah, why speed up just to slam on the brakes? That seems odd.
It does, but it's essential. Remember, the goal isn't just delivery to the area, it's getting the defense materials out of the blood and into the tissue where the problem is.
Ah, right, the exudate formation.
Exactly. That increased vascular permeability, the leakiness we just talked about, starts almost immediately after the vasodilation. So as this protein-rich fluid, the plasma, starts pouring out of the leaky vessels to form the exudate.
The stuff left inside the vessel gets thicker.
Precisely. The blood volume inside the vessel decreases, but the cells, particularly the red blood cells, are mostly left behind. This loss of the watery plasma makes the remaining blood much more concentrated, much more viscous.
It's sludge, almost.
It can be, yeah. So now you have this thick, viscous blood trying to flow through vessels that are actually wider than normal because of the vasodilation.
Wider pipes, thicker fluid, equals slow flow.
Exactly. The flow slows down dramatically. The small vessels become engorged with these slowly moving, concentrated red cells. That's stasis.
Okay, so the traffic jam isn't an accident, it's part of the plan. And this slowing down is absolutely necessary for the next critical step, which involves the white blood cells, the leukocytes. You mentioned leukocyte margination.
Yes. Stasis completely changes the physics inside the vessel. In normal, fast-flowing blood, cells tend to travel in the center of the stream. Laminar flow keeps them away from the walls.
Like logs floating down the middle of a river.
Perfect analogy. But when the flow slows right down during stasis, the heavier elements, especially the white blood cells, primarily neutrophils at this early stage, get pushed out of that central column. They drift towards the edges.
They marginate. They move to the margins.
Exactly. They accumulate along the inner lining of the vessel, the endothelium. This physical process of them lining up along the wall is called margination.
So now they're right up against the wall, perfectly positioned, but they aren't sticking yet, are they? Just touching.
They're basically staged, ready to interact. And crucially, while this is happening, the endothelial cells that make up the vessel wall are also being activated. They're getting signals from the injury site, too.
What are they doing?
They start expressing specialized adhesion molecules on their surface. Think of it like putting out molecular Velcro strips. This prepares the wall for the leukocytes to grab on and eventually exit. Stasis is critical because it gives the marginated leukocytes the time and proximity needed to actually interact with this newly deployed Velcro.
Okay, the stage is set. Slow flow, cells at the edge, walls getting sticky. Now we need the main event, the leak itself. Increased vascular permeability. This is what defines the whole inflammatory exudate.
This is the absolute hallmark vascular change, yes. It allows those large plasma proteins and the leukocytes to move out into the tissue. And where does this primarily happen?
You said postcapillary venules, the
Yeah.
little veins just after the capillaries.
That's the main site. They have thinner walls compared to arterioles, and their endothelial cells are particularly sensitive to the chemical signals that trigger permeability changes. The sources describe two major mechanisms for how these leaks are created, based largely on timing.
Okay, let's see the first one, the quick response. Endothelial cell contraction.
This is a very rapid, active process. It's triggered by chemical mediators. We mentioned histamine, but also things like bradykinin and leukotrienes. These molecules bind to receptors on the endothelial cells and cause the cells to physically contract.
They shrink.
Sort of pull away from each other, creating temporary gaps between the cells, endothelial gaps. These gaps are large enough for plasma proteins and fluid to pour out quickly.
And you mentioned timing is key here. How fast and how long does this last?
It's called the immediate transient response for a reason. It happens almost instantly when the mediator hits, think of a bee sting, that immediate swelling.
Right, it happens right away.
But it's short-lived. Typically, it only lasts about 15 to 30 minutes, maybe an hour max, because the mediators like histamine are rapidly broken down or washed away. It's the quick initial burst of swelling.
Okay, so that's the fast temporary leak. What's the second mechanism? It sounds more serious. Endothelial injury.
It is. This isn't just cells contracting. This is direct damage leading to the death, necrosis, and detachment of the endothelial cells themselves. The vessel lining is actually breached.
What causes that kind of damage?
Severe insults. Think major thermal burns, exposure to really potent microbial toxins, certain chemicals. The damage is direct and physical.
And the timeline for leakage must be completely different then.
Totally different. The leakage starts immediately, just like with contraction, because the barrier is broken. But it's sustained. It can last for hours or even days, continuing until the damaged vessels are somehow repaired or maybe thrombosed, clotted off.
Wow, okay. Much longer lasting.
And interestingly, the inflammatory cells themselves, especially the neutrophils that arrive later, can actually contribute to this secondary wave of injury. They release toxic oxygen radicals and enzymes that can damage bystander endothelial cells, potentially prolonging the leakage.
So the cleanup crew can sometimes make the mess worse, in a way.
It's a double-edged sword, yes. And the source material also mentions a sort of middle ground, a delayed, prolonged leakage. What's that about?
Yeah, I saw that. Delayed but sustained.
You see this with certain milder forms of direct injury. Classic example is sunburn, or maybe some types of radiation exposure or certain bacterial toxins. The leakage doesn't start right away. There's a delay, maybe 2 hours, maybe up to 12 hours.
Why the delay?
It suggests the cell damage isn't instantaneous cell death, but maybe triggers an apoptotic pathway or some slower process that leads to increased permeability later on. But once it starts, this delayed leakage is also sustained for several hours, sometimes even days.
So 3 patterns. Immediate and short, immediate and long, or delayed and long. The body has different ways to spring a leak depending on the insult.
Exactly. It shows a real sophistication in the response. But the net result of all this leakage, regardless of the exact mechanism, is that buildup of high-protein fluid in the tissue.
Which we see and feel as edema, the swelling. That's the tumor cardinal sign.
Precisely.
Now, while all this leaking is happening and the exudate is forming, the local area is basically getting flooded. How does the body deal with all that extra fluid? That brings in the lymphatic system, right? The drainage network.
Absolutely crucial. We often focus on the blood vessels, but the lymphatics are working overtime during acute inflammation. They act as the essential emergency drainage system.
What exactly are they doing in this acute phase?
Several key things. First, lymph flow itself is dramatically increased. It has to be, to handle the sheer volume of edema fluid pouring out from the leaky blood vessels.
Just trying to drain the swamp.
Pretty much. Second, they don't just drain fluid, they also transport the larger stuff that can't easily get back into the blood capillaries. Things like leaked plasma proteins, cellular debris from the injury, the inflammatory cells themselves, and, critically, any microbes or antigens.
Ah, so they're not just storm drains. They're also carrying the evidence, the microbes, away from the site.
Exactly. They transport it to the regional lymph nodes, which are like the local police stations or intelligence hubs of the immune system. This connects the site of inflammation to the broader adaptive immune response.
And the sources mention this process can be so active that it causes its own visible problems, especially with bacterial infections.
Yes, you can get lymphangitis and lymphadenitis.
Okay, break those down. Lymphangitis.
That's when the lymphatic vessels themselves become inflamed, usually because bacteria are actively spreading along them. Clinically, this is what causes those characteristic red streaks you sometimes see on the skin leading away from an infected wound. It's literally tracing the inflamed superficial lymph vessel.
Wow, okay, that's a serious sign then.
It is. And lymphadenitis is what happens at the destination, the draining lymph node. It becomes swollen, tender, painful.
Why does it swell?
Because it's going into overdrive processing all the incoming fluid, debris, microbes, and activated immune cells arriving via the lymph. The lymph node undergoes reactive hyperplasia, the internal structures, the lymphoid follicles enlarge, and then the number of lymphocytes and macrophages inside increases dramatically as it mounts an immune response.
That's a really powerful clinical connection. The local signs, redness, heat, swelling are right there,
Right.
but those red streaks or a swollen, painful node nearby tell you the battle has expanded. The infection might be spreading through the body's transport network.
Precisely. It links the local vascular events to the systemic immune surveillance.
Okay, so the vessels have slowed the flow, stasis, opened the gates, permeability, and the leukocytes are lined up along the wall, margination. Now we need to follow those leukocytes, the defense cells, as they actually get out. This is a multi-step journey, right? Highly choreographed.
Extremely choreographed. It's a beautiful cascade driven by molecular signals. And it has to be specific. The body doesn't want just any cell popping out anywhere. It needs the right cell, usually neutrophils first, then macrophages, at the specific site of injury. We can break this journey down into 3 main phases.
Right. Phase 1, still inside the blood vessel in the lumen. This involves rolling and adhesion. What's happening here?
So the first contact, that initial sticky interaction between the marginated leukocyte and the vessel wall, is rolling. It's a very transient binding.
Rolling, like a ball.
Almost, yeah. It's mediated by a family of adhesion molecules called selectins. Think of them as providing low-affinity connections. L-selectin is on the leukocyte surface, while E-selectin and P-selectin are expressed on the activated endothelial cell surface.
Low affinity meaning they grab on but let go easily.
Exactly. Low affinity, fast off rate. That's what creates the characteristic tumbling or rolling motion along the endothelial surface. The cell briefly sticks, detaches, tumbles forward, sticks again.
And the appearance of these selectins on the endothelium is triggered by the inflammatory signals.
Oh, absolutely. It's tightly regulated. Pro-inflammatory cytokines, the master regulators like TNF and IL-1 that we'll get to, induce the endothelial cells to start synthesizing and expressing E-selectin. That takes an hour or 2.
Okay, that's fairly quick.
But there's an even faster mechanism for P-selectin. Mediators like histamine and thrombin cause the rapid mobilization of P-selectin that's already preformed and stored inside the endothelial cells in little packages called Weigel-Pallade bodies.
So they have emergency P-selectin ready to go?
Instantly deployable, like throwing out sticky nets onto the vessel wall within minutes or even seconds of the initial trigger. This ensures that rolling can begin almost immediately.
Okay, so rolling starts fast, but rolling isn't getting out. The cell needs to stop and grab on tightly, right? That's the firm adhesion part.
Correct. Rolling slows the cell down and allows it to scan the surface. Firm adhesion is the next step, where the cell comes to a complete halt and latches on securely. This involves a totally different class of adhesion molecules, the integrins.
Integrins. I remember these are interesting because they act like molecular switches. They're usually off or low stickiness, but something flips them to on or high stickiness.
That's the key concept. Integrins like LFA1 and MAC1 on the leukocyte surface normally exist in a low affinity state. They don't bind strongly to their partners on the endothelium.
What flips the switch? What makes them sticky?
Chemokines. These are specific chemotracting chemicals. We'll discuss them more with mediators that are produced at the injury site and then displayed on the surface of the activated endothelial cells bound to proteoglycans.
So the rolling leukocyte bumps into these chemokines.
Yes. As the leukocyte rolls along, chemokine receptors on its surface bind to these displayed chemokines. This binding triggers a rapid inside-out signaling cascade within the leukocyte. Within seconds, this signal converts the integrins on the leukocyte surface from their default low-affinity state to a high-affinity state.
That's the commitment signal. Now they grab on tight.
They grab on very tightly to their specific ligands on the endothelial surface molecules like ICAM1 and VCAM1, whose expression is also upregulated by TNF and IL-1.
Okay.
So the leukocyte stops rolling dead in its tracks. It adheres firmly, spreads out, flattens against the endothelium, reorganizes its internal structure, its cytoskeleton, and gets ready for the next phase, squeezing through the wall.
Phase 2, migration across the endothelium. This is the actual escape, often called diapedesis or transmigration. How do they do that? Squeeze between the endothelial cells.
Yes, primarily. Once they're firmly adhered, the leukocytes actively migrate between the endothelial cells, finding those interendothelial junctions that might have already been slightly widened by histamine or are just naturally potential exit points.
Where does this usually happen? Still the post-capillary venules.
Predominantly, yes. Still the postcapillary venules. They are essentially crawling towards the source of the chemokines that triggered their firm adhesion in the first place following the chemical trail.
Is there a specific molecule that helps them navigate these junctions? It seems like a tight squeeze.
It is a tight squeeze, and it requires active movement from the leukocyte. But there are molecules that help facilitate it. A key one mentioned is CD31, also known as PCAM1. It's expressed on both the leukocyte and the endothelial cells, particularly at the intercellular junctions, and interactions between them seem to help guide the leukocyte through.
Okay, so they wiggle through the endothelial layer. What about the layer underneath? The basement membrane.
Good point. After clearing the endothelial cells, they encounter the basement membrane, which is a denser layer of extracellular matrix. They have to breach that too. It's thought they achieve this by secreting enzymes like collagenases and other proteases right at the point of contact. These enzymes locally digest a small path through the basement membrane, allowing the leukocyte to fully exit the vessel and enter the interstitial tissue.
All right, phase 3, they're out. They're in the tissue now. But the actual site of injury or the bacteria might still be some distance away. So the final step is directed movement within the tissue, right? Chemotaxis.
Exactly. Chemotaxis is simply locomotion oriented along a chemical gradient. The leukocyte needs a navigation system to find the hot spot.
A molecular GPS. What provides the signal? What are the chemical beacons they follow?
There are two main categories of chemoattractants. First, you have exogenous ones, meaning they come from outside the body. The classic example is bacterial products. Many bacteria release small peptides, particularly those starting with a specific amino acid sequence, N-formylmethionine, which our leukocytes have receptors for. They can literally smell the bacteria.
Wow, okay, so they follow the scent of the invaders. What about signals from our own body, endogenous ones?
Yes, several crucial endogenous chemoattractants are generated during inflammation. 1. Chemokines. We already mentioned these. Specific chemokines like IL-8 or CXCL8 are powerful attractants, especially for neutrophils. 2. Complement components. Parts of the complement system, another defense cascade. The fragment C5a is a particularly potent chemoattractant for many leukocytes. 3. Arachidonic acid metabolites. Products derived from lipids. Specifically, leukotriene B4, LTB4, is a major chemoattractant, again, especially for neutrophils.
So the body sets up multiple overlapping chemical trails leading to the site of trouble. How does the cell physically move along this trail? It's not just drifting, right? It's actively crawling.
Highly active crawling, yes. The leukocyte has receptors all over its surface for these chemoattractants. When the receptors on the front end of the cell bind more chemoattractant molecules than the receptors on the back end, it triggers internal signaling.
What does the signaling do?
It causes a dramatic reorganization of the cell's internal skeleton, the cytoskeleton. Specifically, it triggers the polymerization of actin filaments at the leading edge, pushing the membrane forward in projections called pseudopods or lamellipodia. At the same time, myosin motors at the back of the cell contract.
So it extends the front and pulls up the rear.
Essentially, yes. It develops this sort of front-wheel drive mechanism, actively pulling itself along the substrate, the extracellular matrix, towards the increasing concentration of the chemoattractant signal until it reaches the source.
Now let's talk about the timing of which cells arrive, because this is a classic finding in pathology labs. The type of cell tells you how old the inflammation is. Why do neutrophils absolutely dominate that early phase?
Neutrophils are the first responders, hitting the scene in large numbers within the first 6 to 24 hours. There are 3 main reasons for this dominance.
Okay, what are they?
1. Numbers. They are simply the most abundant type of circulating white blood cell. There are lots of them ready to go. 2. Speed of response. They respond very quickly and strongly to the initial wave of chemo.
But why are they better later on?
Several reasons. 1. Longevity. Macrophages can survive in the tissues for much longer periods, weeks, months, even years. 2. Proliferation. Unlike neutrophils, macrophages can actually divide and proliferate within the inflamed tissue, increasing their numbers locally. 3. Function. While neutrophils are primarily killers and phagocytes, macrophages are much more versatile. They are powerhouse phagocytes, cleaning up dead neutrophils, cellular debris, and pathogens, but they also play a crucial role in orchestrating the repair process. They release growth factors that stimulate tissue rebuilding and angiogenesis.
So they shift from just fighting the fire to also cleaning up the damage and starting reconstruction.
Precisely. So that shift from a neutrophil-dominated infiltrate in the first day or so to a macrophage-dominated one after 48 hours is a key indicator. It often signals either that the initial injurious agent is still present, requiring a sustained response, or that the process is transitioning towards resolution and repair, or potentially, if the stimulus persists indefinitely, towards chronic inflammation.
Okay, this entire incredibly complex dance, the vessels changing shape, the cells rolling, sticking, squeezing out, crawling to the target, none of this happens spontaneously. It's all directed by a huge array of chemical messengers, the mediators of inflammation. Let's dive into these chemical orchestrators. Where should we start? Maybe with the fastest acting ones.
That makes sense. The ones that are ready to go instantly, that really means the vasoactive amines.
And the main player there is histamine.
Absolutely. Histamine is the quintessential immediate mediator. It's preformed and stored in granules, primarily within mast cells. And mast cells are strategically positioned right next to blood vessels and connective tissues throughout the body. Basophils and platelets also contain histamine.
Locked and loaded. What triggers its release?
A variety of stimuli: physical injury trauma, heat, cold, immune reactions, particularly allergic reactions where IgE antibodies bind to mast cells. And importantly, certain products of the complement system, specifically the fragments called anaphylatoxins, C3A and C5A, are potent triggers for histamine release.
And its action is immediate.
Instantaneous. It causes dilation of arterioles and, crucially, that rapid contraction of endothelial cells in venules, creating the gaps for the immediate transient increase in vascular permeability. It's the principal driver of that very first wave of vasodilation and leakiness.
Okay, histamine is the first alarm. The sources also briefly mention serotonin. Where does that fit in?
Serotonin is another vasoactive amine found mainly in platelet granules. Its role in human inflammation seems less prominent than histamines. It can cause vasoconstriction or vasodilation depending on the context, but it's generally considered a less critical player in the typical acute inflammatory vascular response compared to histamine.
Right, moving from the preformed amines to mediators that have to be synthesized on the spot. This brings us to the arachidonic acid AA metabolites. These sound important, especially for symptoms like pain and fever.
Hugely important. These are lipid mediators, collectively called eicosanoids. They are synthesized de novo, made fresh when needed from arachidonic acid, which is a fatty acid component of cell membrane phospholipids. When a cell is activated or injured, enzymes like phospholipase A2 release AA from the membrane.
And then AA goes down different pathways. Let's start with the cyclooxygenase COX pathway. This makes prostaglandins, PGs, right?
Yeah.
And there are two COX enzymes.
Correct. There's COX1 and COX2. COX1 is considered the housekeeping enzyme. It's constitutively expressed in most tissues and produces prostaglandins involved in normal physiological functions like protecting the stomach lining and regulating kidney blood flow.
And COX2.
COX2 is the inducible enzyme. Its expression is normally low, but it gets massively upregulated by inflammatory stimuli, cytokines like TNF and IL-1, growth factors, etc. So COX2 is primarily responsible for producing the prostaglandins that drive inflammation. These PGs are synthesized by various cells, including mast cells, macrophages, endothelial cells.
What do these inflammatory prostaglandins do?
They have multiple roles. Several of them, like PGD2, PGE2, and PGI2, prostacyclin, are potent vasodilators and also contribute to the increased vascular permeability, synergizing with histamine and other mediators to promote edema.
Okay, vasodilation and leakiness. What about pain and fever?
That's largely PGE2. PGE2 doesn't directly cause pain itself, but it makes the area exquisitely sensitive to other pain-provoking stimuli, like bradykinin. It's highly hyperalgesic. And critically, PGE2 acts on the hypothalamus in the brain to reset the body's thermostat, causing the systemic fever response.
So that's why COX inhibitors like aspirin or ibuprofen reduce fever and pain. They block PGE2 synthesis.
Exactly. Now, within the COX pathway, there's a really critical balance we need to highlight, especially with clinical relevance. That's the balance between PGI2, prostacyclin, and TXA2, thromboxane A2.
Right, you mentioned this earlier. What's the difference?
PGI2, or prostacyclin, is mainly produced by healthy vascular endothelial cells, using KIOX2, interestingly. It's a powerful vasodilator and, very importantly, a potent inhibitor of platelet aggregation. It helps keep blood flowing smoothly and prevents clots from forming where they shouldn't.
Okay, PGI2 is anti-clotting, co-flow. What about TXA2?
TXA2, or thromboxane A2, is primarily produced by activated platelets using COX1. It does the exact opposite. It's a potent vasoconstrictor and strongly promotes platelet aggregation. It's essential for forming a blood clot when you have an injury.
So a natural push-pull system. Healthy endothelium makes PGI2 to keep things open, but activated platelets make TXA2 to plug leaks.
Precisely. The balance between PGI2 and TXA2 is absolutely critical for maintaining vascular homeostasis and regulating thrombosis. And disrupting this balance is key to understanding some drug side effects, which we'll get to.
Okay, that's the COX pathway, prostaglandins, pain, fever, and that PGI2 TXA2 balance.
Yeah.
What about the other major route for arachidonic acid, the lipoxygenase pathway?
This pathway generates a different set of lipid mediators, mainly the leukotrienes, LTs, and the lipoxins, LXs. The key enzyme here for leukotrienes is 5-lipoxygenase, which is found predominantly in leukocytes, especially neutrophils and mast cells.
What do the leukotrienes do? Are they also involved in vascular changes?
Yes, but they also have major roles in cell recruitment. Let's break them down. First, there's leukotriene B4, LTB4.
LTB4, what's its main job?
LTB4 is one of the most potent chemotactic agents known, particularly for neutrophils. It's a powerful signal calling neutrophils to the site of inflammation. It also activates neutrophils, getting them ready for phagocytosis and killing.
So LTB4 is all about recruiting the troops. What about the other leukotrienes? The sources mention cysteine LTs.
Right. These are LTC4, LTD4, and LTE4. They are called cysteine LTs because they have cysteine-containing peptides attached. Unlike LTB4, their main effects are vascular and smooth muscle related. They cause intense vasoconstriction, increase vascular permeability, often more potently and longer lasting than histamine, and perhaps most notoriously, cause significant bronchospasm contraction of airway smooth muscle.
Ah, sounds like asthma.
Exactly. The cysteine LTs are major players in the pathophysiology of asthma and allergic reactions. Drugs that block their synthesis or their receptors are used to treat asthma.
So LTs, LTB4 recruit cells, the cysteine LTs cause leakiness and spasm.
Right.
Now, you also mentioned lipoxins. LXs come from this pathway. What are they? They sound different.
They are very different. Lipoxins are actually the stop signals or resolution mediators. They're also derived from arachidonic acid, but their synthesis is more complex, often requiring interaction between two different cell types, like neutrophils making an intermediate, and then platelets converting it to the active lipoxin.
And their function is to stop inflammation.
Yes, they actively suppress inflammation. They inhibit neutrophil chemotaxis and adhesion to the endothelium. They essentially put the brakes on the acute inflammatory response and help initiate the transition towards resolution and cleanup. They counteract the pro-inflammatory PGs and LTs.
Okay, we've covered the amines and the lipid mediators. Now for the protein messengers, cytokines and chemokines. These sound like major controllers. Let's start with the big bosses, TNF and IL-1.
Tumor necrosis factor, TNF, and interleukin-1, IL-1, are indeed master regulators, especially in the early phases. They are produced mainly by activated macrophages and dendritic cells, but other cells can make them too, often in response to bacterial products or immune signals.
If histamine is the first shot, what do TNF and IL-1 do?
Their primary role in acute inflammation is endothelial activation. They are the main signals that tell the endothelial cells lining the blood vessels to express those adhesion molecules, the selectins, E-selectin, and the ligands for integrins like ICAM1, VCM1.
So they make the vessel wall sticky for the rolling and adhesion phases.
Precisely. Without TNF and IL-1, leukocytes wouldn't adhere effectively and wouldn't get out. They also powerfully amplify the whole response by stimulating endothelial cells and leukocytes to produce other mediators, including more cytokines, chemokines, and importantly, inducing COX-2 for prostaglandin synthesis.
They turn up the volume on everything else.
Pretty much. And they also have major systemic effects. They are the key drivers of the systemic acute phase response that makes you feel sick, fever, acting on the hypothalamus, lethargy, loss of appetite. They also stimulate the liver to produce acute phase proteins.
And if they stick around too long.
If TNF production becomes chronic and sustained, it can lead to severe metabolic problems, including insulin resistance and muscle wasting known as cachexia, which is seen in some chronic inflammatory diseases and cancers.
Okay, so TNF and IL-1 are the master activators. What about chemokines? We mentioned them in leukocyte recruitment.
Chemokines are a large family of small proteins whose main function is just what their name suggests: chemoattraction plus cytokine. They act primarily as chemoattractants to guide the migration of specific types of immune cells.
Traffic controllers.
Exactly. We can broadly divide them. Inflammatory chemokines, like IL-8, CXCL8, are produced rapidly at sites of infection or injury, often induced by microbes or TNF-IL-1. Their main job is to recruit leukocytes, particularly neutrophils, from the blood into the tissues.
And how do they do that recruitment? Just by providing a gradient.
The gradient guides the chemotaxis in the tissue, yes. But critically, on the endothelium, Binding of chemokines, like IL-8, to receptors on the rolling leukocyte is what triggers that rapid increase in integrin affinity, leading to firm adhesion. So they are essential for both stopping the cells and telling them where to go.
Are there other types of chemokines?
Yes, there are also homeostatic chemokines involved in organizing lymphoid tissues and guiding lymphocyte trafficking under normal conditions. But the inflammatory ones are key here. Some, like fractokin, CX3CL1, exist in both membrane-bound and soluble forms and promote strong adhesion of monocytes and T cells.
Okay, one more major system involved, the complement system. This is that cascade of plasma proteins. How does it plug into the vascular reaction?
Complement is a powerhouse contributor. It's a cascade of plasma proteins that can be activated in several ways by antibodies binding to a microbe, classical pathway, directly by microbial surfaces, alternative pathway, or by mannose binding nectin, lectin pathway. But they all converge.
Where do they converge?
They all lead to the cleavage of the central component, C3, into two key fragments, C3a and C3b. C3b can then lead to the cleavage of C5a into C5a and C5b.
In these fragments, C3a and C5a are the inflammatory mediators?
Yes, along with C4a produced earlier in some pathways. These small fragments, C3a, C4a, and especially C5a, are collectively called anaphylatoxins.
Why anaphylatoxin?
Because they cause effects similar to anaphylaxis, they directly stimulate mast cell degranulation, leading to the release of histamine and other mediator.
Ah, so complement activation feeds directly back into boosting histamine release, causing more vasodilation and permeability.
Exactly. It's a powerful amplification loop. And C5a is particularly potent and versatile. Not only is it an anaphylatoxin, but it's also one of the most powerful chemoattractants for neutrophils, monocytes, eosinophils, and basophils. It also directly activates leukocytes, stimulating the arachidonic acid pathway within them, leading to more leukotriene production.
So complement does three things.
Okay.
Punches holes in microbes via the later C5b-C9 complex, opsonizes them for phagocytosis via C3b, and directly fuels the vascular inflammatory response and cell recruitment via C3a and C5a.
You got it. It's a multifunctional weapon system.
Okay, one last group of mediators mentioned. The kinins, what are they?
The kinin system is another plasma protein cascade system. The main end product is bradykinin.
Bradykinin, what does it do?
It's similar in action to histamine, actually. It increases vascular permeability, causes contraction of smooth muscle, though vasodilation in arterioles, and importantly, it's one of the main mediators that directly stimulates pain nerve endings. It's a major contributor to the sensation of pain, dolor, during inflammation. It's usually short-lived because it's rapidly inactivated by enzymes called kininases.
Wow. Okay, that is an incredibly complex orchestra of chemical signals, amines, lipids, proteins, cascades, all working together to drive these vascular changes in cell movements. Understanding this detail, I guess, is absolutely fundamental for developing drugs to control inflammation.
It's the entire basis for rational anti-inflammatory therapy. Almost every drug we use targets one or more steps in these pathways we've just discussed.
Let's start with the most common ones, targeting those arachidonic acid metabolites, the eicosanoids, the NSAIDs, non-steroidal anti-inflammatory drugs, aspirin and ibuprofen. How do they work?
Their mechanism is relatively simple but profound. They inhibit the cyclooxygenase COX enzymes, both COX-1 and COX-2, to varying degrees depending on the specific drug.
So they block the production of prostaglandins.
Exactly. By blocking PG synthesis, especially PGE2, they effectively reduce fever by preventing PGE2 action on the hypothalamus, and pain by preventing PGE2's hyperalgesic effect. They also have anti-inflammatory effects by reducing PG-mediated vasodilation and edema, though they don't hit all aspects of inflammation.
Now, the sources highlighted the story of the selective COX-2 inhibitors. The idea was great, right? Target the bad, inflammatory COX-2, spare the good, housekeeping COX-1. Why did that run into problems?
It ran into problems because of that delicate PGI2-TXA2 balance we talked about. Remember, TXA2, the pro-clotting vasoconstrictor, is made mainly by platelets using COX-1. PGI2, the anti-clotting vasodilator, is made mainly by endothelial cells, largely using COX-2.
Ah, I see where this is going.
When you selectively inhibit only COX-2, you significantly reduce the production of the protective PGI2 by the endothelium, but you leave the COX-1 pathway and platelets untouched. So TXA2 production continues unchecked.
You remove the brake, PGI2, but leave the accelerator, TXA2, fully functional.
Exactly. You tip the balance heavily towards vasoconstriction and platelet aggregation. This imbalance is thought to be the primary reason why several selective COX2 inhibitors were associated with an increased risk of cardiovascular events, like heart attacks and strokes. It's a stark lesson in the unintended consequences of highly specific targeting in a complex biological system.
A really powerful example. What about the big guns, when you need broad suppression? Corticosteroids.
Corticosteroids are incredibly potent and broad-spectrum anti-inflammatory agents. They don't just target one enzyme, they work primarily by affecting gene transcription. They bind to intracellular receptors, and this complex then influences the expression of numerous genes.
What genes do they suppress?
They suppress the expression of many pro-inflammatory genes, including those encoding COX2, phospholipase A2, the enzyme that releases AA in the first place, and key cytokines like TNF and IL-1, as well as adhesion molecules. They basically turn down the volume on the entire inflammatory orchestra at multiple levels.
Which makes them effective, but also explains their wide range of side effects if used long-term.
Precisely.
We're also seeing therapies that are much more targeted at the protein level, like the TNF antagonists. How successful have they been?
They've been revolutionary for treating certain chronic inflammatory diseases like rheumatoid arthritis, inflammatory bowel disease, and psoriasis, where TNF is a major driver of the pathology. By specifically blocking TNF, you can often achieve dramatic improvements.
But again, there's a trade-off.
Always a trade-off. TNF is critical for normal host defense, especially against intracellular pathogens like mycobacteria, which cause tuberculosis. Blocking TNF significantly increases the risk of serious infections. It highlights that inflammation, even when causing disease, evolved for a vital protective purpose.
And briefly, there are also drugs targeting cell movement itself,
Yeah.
like integrin antagonists.
Yes, drugs that block specific integrins involved in leukocyte adhesion are used or being tested for conditions like multiple sclerosis and inflammatory bowel disease, aiming to prevent the damaging influx of leukocytes into tissues. And even things like dietary manipulation, increasing omega-3 fatty acids from fish oil, for instance, can subtly shift arachidonic acid metabolism away from pro-inflammatory PGs and LTs towards more anti-inflammatory mediators like resolvins and protectins, which actively promote resolution.
So pulling this all together after this deep dive, acute inflammation really emerges as this incredibly rapid, tightly controlled, chemically driven process. It's all about logistics, moving defense materials like fluid, proteins, and cells for the bloodstream into the exact tissue location where they're needed.
Absolutely. And when you understand the underlying vascular and cellular events, those classical cardinal signs make perfect sense. Redness, rubor, and heat, calor. That's the direct result of vasodilation and increased blood flow, kicked off mainly by histamine and prostaglandins. Swelling, tumor, that's the physical manifestation of increased vascular permeability and the formation of the protein-rich exudate. Pain, dolor, that's the sensory consequence of chemical mediators like bradykinin and PGE2 sensitizing local nerve endings.
And the fifth sign, loss of function, often follows from the pain and swelling. What are the potential outcomes after this whole reaction happens? Does it always just fix the problem?
The ideal outcome and the goal is complete resolution. The injurious agent is eliminated, the mediators break down, the vascular changes reverse, the fluid drains, the cells leave or die off, and the tissue returns to normal structure and function.
But that doesn't always happen.
No. If the initial injury caused substantial tissue destruction, or if the inflammation occurs in a tissue that can't regenerate easily, the outcome might be scarring or fibrosis. The defect gets filled in with connective tissue instead of functional tissue.
And the third possibility.
If the injurious agent persists, maybe it's a microbe the body can't eliminate, or a persistent toxin or an autoimmune reaction, the acute inflammatory response might fail to resolve and instead transition into chronic inflammation. This involves a different set of cells, lymphocytes, macrophages, ongoing tissue injury, and simultaneous attempts at repair, often leading to long-term damage.
Okay, we spent a lot of time today on the incredible complexity and the delicate balances involved, PGI2 versus TXA2, the risks of blocking TNF. It really underscores how vital yet potentially dangerous inflammation is. This leaves us with a final thought to ponder.
Given that inflammation is absolutely essential for survival, we can't just eliminate it. And given the serious side effects we see with broad suppression, like with corticosteroids or even TNF blockers,
What if we could be more surgical? Could we develop therapies that specifically target just one aspect, like the pathological increase in vascular permeability, the leakiness that causes so much tissue damage, but do it in a highly localized way? Could we stop the damaging edema without blocking the necessary arrival and function of the leukocytes needed to clear an infection?
that is precisely the challenge and perhaps the holy grail of future anti-inflammatory drug development. Can we find signaling pathways within the endothelial cell, for instance, that are uniquely activated during excessive or inappropriate leakage, but aren't required for the baseline trafficking of immune cells or the necessary response to a genuine threat?
Achieving that level of specific, localized control over the leak without compromising essential defense.
That would be transformative. It requires a much deeper understanding of the endothelial cell's internal signaling during different types of inflammatory stress. It's definitely a fascinating area for ongoing research and certainly something for you, the listener, to think about further.